Hello, everyone. My name is Sai Sadna, and today I will be presenting about neuromuscular junction and excitation, which includes the contraction and coupling of muscle. To start off with, I will be going over the objectives of this presentation and the topics we will be focusing on. The first one would be we would look into how to draw and label a neuromuscular junction. Then we will move on to the steps at the NMJ leading to the muscle cell excitation. After which, we will be outlining the sequence of events in the skeletal muscle cell excitation. We will then be focusing on the definition and what excitation contraction coupling actually is. And last but not the least, we will be predicting the effects of protein malfunctions in the process of NMJ. So to start off with, the neuromuscular junction, what actually it is. So a NMJ is a chemical synapse between an efferent neuron of the somatic motor division and a skeletal muscle cell. So it allows the electrical signal of a neuron to be translated to a neurotransmitter that will subsequently excite the skeletal muscle cell, resulting in a contraction process. As you can see in this figure, the main labels that we do have to focus on would be the presynaptic terminal, the synaptic vesicles, the synaptic cleft, the postsynaptic membrane, and the sarcolemma, which is basically a muscle fiber membrane. So these are the main labels that we will be focusing on. It will be easier to understand this figure the more and more we get deeper into the topics and the steps of the NMJ. So the NMJ takes place with a lot of steps in process, and there are 6 main steps that we will be focusing on. The first one is the action potential arrival at the axon terminal. So what this actually means is that the process begins with an action potential or an electrical impulse traveling along the axon, as you can see here, of a motor neuron. The action potential is generated by the movement of sodium and potassium ions across the neuron's membrane, creating a wave of electrical charge that propagates down the axon, which we can clearly see even though that the sodium and potassium ions are not labeled here, but these are the charges that move down the axon terminal. Now, once this happens, we then have a calcium influx that goes into the axon terminal. When the action potential reaches the axon terminal, it causes the voltage-gated calcium channels to open. That is exactly what is depicted here. As a result, calcium ions flow into the axon terminal from the extracellular fluid due to higher concentration of calcium outside the neuron, which is basically here, as you can see, there's calcium influx in the extracellular fluid right here, and then it goes into the axon terminal. When this happens, we then have an ACH release into the synaptic cleft. So the influx of calcium ions triggers synaptic vesicles that we spoke about in the first figure, filled in with acetylcholine, which we're going to call as ACH, to move towards and fuse with the presynaptic membrane. Now, this process is called exocytosis. It releases the ACH into the synaptic cleft, which is the space between the axon terminal and the muscle fiber membrane. As I said, once the second step is over, the synaptic vesicles that are right here, they have ACH filled in them because of the influx of calcium that we just saw. Now, once this happens, the ACH is then into the synaptic cleft and also the sarcolemma. After this, we then have the ACH binding to the receptors on the motor end plate. So the ACH molecules diffuse across the synaptic cleft and bind to nicotinic acetylcholine receptors on the motor end plate, the specialized region of the muscle fiber membrane. These receptors are ligand-gated ion channels. Generation of end plate potential. After this happens, we then have the generation of the end plate potential. So the binding of the ACH to its receptors causes the ligand-gated ion channels to open. And these ligand-gated ion channels are the ones that we spoke about that happens in the motor end plate. So this then allows sodium ion to flow into the muscle fiber and a small amount of potassium to flow out. This ion movement depolarizes the motor end plate, creating a local change in membrane potential called the end plate potential, EPP, that we will be using. If the EPP is strong enough, it will trigger an action potential in the muscle fiber. Once this is done, the action potential in the muscle fiber reaches the threshold. So if the EPP reaches the threshold, it initiates an action potential that propagates along the muscle fiber, which is the sarcolemma, and down the T tubules. This action potential leads to the release of calcium from the sarcoplasmic reticulum, ultimately causing muscle contractions. Now, these are the steps that happen in a proper order for a neuromuscular junction to happen. Now, how does this lead to an excitation of the skeletal muscle cell, which thereby leads into a contraction and coupling? So, the action potential that we just spoke about propagates along sarcolemma and T tubules. So, the action potential generated at the neuromuscular junction spreads along the muscle fiber membrane, which is the sarcolemma, and travels down the T tubules, which is the transverse tubules. The T tubules are invaginations of the sarcolemma that penetrate deep into the muscle fiber, allowing the action potential to quickly reach the interior of the cell. We then have the calcium release from the sarcoplasmic reticulum. The action potential in the T tubules triggers voltage-sensitive DHBR, which is the dihydropyridine receptors, to activate ryanodine receptors on sarcoplasmic reticulum. So this causes the SR to release stored calcium ions into the cytoplasm of the muscle fiber. We then have the calcium binding to troponin. So the released calcium that just happened, it binds into the troponin, a regulatory protein on the thin filaments of the muscle fiber. This binding causes a conformational change in troponin, which moves tropomyosin away from the myosin binding sites on the actin filament. We then have the cross-bridge cycling. So the cross-bridge formation is basically with the tropomyosin moved out of the way, the myosin heads can bind to now exposed myosin binding sites on actin filaments, forming cross-bridges. Once this happens, we have the power stroke of myosin heads. On the cross bridge are formed the myosin heads pivot, pulling the actin filament towards the center of the sarcomere. This action is known as power stroke and is powered by the hydrolysis of ATP. We then have the ATP binding and cross-bridge detachment. After the power stroke, a new ATP molecule binds to the myosin head, causing it to release the actin filament. The myosin head then hydrolyzes the ATP to ADP and pi, which recocks the myosin heads, re-dyeing, readying it for another cycle. We then, at last, have the muscle relaxation. When the action potential sees its calcium is actively pumped back into the SR by calcium ATPase pumps. The decrease in the cytoplasmic calcium causes troponin and tropomyosin to return to their resting positions, blocking the myosin binding sites on actin and leading to muscle relaxation. Now, this is the process of the excitation and for a skeletal muscle cell that we just spoke about. What is excitation contraction coupling? So it is a physiological process that links the electrical excitation of a muscle fiber to its contraction. The excitation of muscle fiber is the process that we just looked into. This process ensures that the electrical signal, which is the action potential, is translated into mechanical response. There are key steps in this. We have the action potential, we have the calcium release, we have the cross-bridge cycling, and we have the muscle fiber shortening. The action potential generated at the NMJ propagates along the muscle fiber, which is the sarcolemma, and into the T tubules. We then have the calcium release. So the action potential triggers the release of calcium ions from the sarcoplasmic reticulum into the cytoplasm of the muscle fiber. Then we have the cross-bridge cycling. Calcium ions bind to troponin, causing a conformational change that moves tropomyosin away from the myosin binding sites on actin filaments. The myosin heads then bind to these sites, forming cross bridges and initiate the power stroke to pulse the actin filaments towards the center of the sarcomere. Muscle fiber shortening. The repeated cycling of cross bridges leads to the shortening of the sarcomeres, which results in the muscle contraction. When the action potential ceases, calcium is bumped back into the SR, leading to a muscle relaxation. Now that we looked into the process of excitation, contraction, and coupling, we will look into the protein malfunction and their effects. So, as the protein, we have the ACH receptors. So malfunctioning ACH receptor at the neuromuscular junction can lead to condition like myasthenia gravis, where the muscle response to the nerve signals is significantly weakened, which leads to muscle fatigue or weakness. Voltage-gated calcium channels in the neuron can lead to impaired ACH release and weak contraction. Dysfunctional calcium channels in the neuron can result in insufficient release of ACH into the synaptic cleft, leading to weak muscle contractions. Ryanodine receptors in the SR. If ryanodine receptors in the sarcoplasmic reticulum malfunction, the release of calcium ions into cytosol is impaired, leading to muscle fatigue due to insufficient calcium availability to contraction. Troponin and tropomyosin. Abnormalities in troponin or tropomyosin can prevent proper binding of myosin to actin, resulting in muscle rigidity and inability to contract. The ATP availability is very important. Insufficient ATP or dysfunctional myosin head disrupts the cross-bridge cycling process, causing muscle fatigue and decrease in contractile force. So this presentation focused on what an NMJ is, what is the process that's involved in the NMJ leading to muscle cell excitation, and the events that take place for this muscle cell excitation to take place in order, and defining the process of contraction and coupling, and what are the protein deficits can lead to malfunctions in the NMJ process.